Hello guys. Welcome to our summer semester histology seminars. So again, we will be doing weekly seminars, and unfortunately for this week, this had to be pre-recorded because we had different schedules as well as you guys had the introductory seminar, so it would have been too many things. So before I begin, I want to talk about like a slightly different approach we will be taking towards these seminars this semester. This is because for this semester you will be looking at slides, systematically, and you have to like identify the slides, do differential diagnostics, and it is more of like special histology than general histology. Last semester it was pretty chill in my opinion, but this semester like Fingerland has to be your best friend, and you have to like be focused in your practical classes and look through the slides constantly. So what we have decided to do is that we will teach you how to identify these slides and teach you how to study with these slides, and give you points in between, like how professors like to ask you questions from the slides, not us like just teaching you theory, because everyone can learn, theory at their own pace. So, without any further delays, let's begin. So, just a basic introduction for the structure of the digestive tube. So, the basic structure of the digestive tube is it is made up of four layers. We have tunica mucosa, tunica submucosa, then we have tunica muscularis externa, and then we have the tunica serosa. Now, this is just the basic structure, which basically means is that it is present in all of the structures. Okay, these layers are present in all the structures of the digestive tube. So first of all, there are three main components of the tunica mucosa. That is the epithelium or the epithelial layer. Then you have the lamina propria mucosa, and then you have the lamina muscularis mucosae. So epithelium, it can be either be the stratified squamous non-keratinized epithelium, or it can be simple columnar epithelium. Now, when you talk about the stratified squamous non-keratinized epithelium, it is mainly present only in esophagus and anal canal, and simple columnar is present in the rest of the digestive tube. When you talk about the lamina propria mucosa, LPM, it just contains loose connective tissue with blood vessels. So LL, you can just remember that. Then we have the lamina muscularis mucosae. It is made up of two layers of smooth muscle. It has inner circular layer and outer longitudinal layer. What this basically refers to is the arrangement of the muscles. Now, when we talk about the esophagus specifically, which I'll explain again next, in esophagus there's only one layer of longitudinal muscle in the lamina muscularis mucosa layer. Now, moving on to the tunica submucosa, it only has loose connective tissue with blood vessels as well as nerves, and it also has submucosal plexus, okay, which is also called Meissner submucosal plexus. It is basically a network of nerve fibers. So now, when we look at these images, you can see the tunica mucosa. It is the most innermost layer. So this top layer would be the epithelium, and here you would have the lamina propria mucosa, and then this would be the lamina muscularis mucosa as it is labeled here. And then below that you have the tunica submucosa, and then these this layers combined is called the muscularis externa. And then sorry, and then when we yeah, then when we look at another angle, as you can see, this is the mucosa, top layer, submucosa, and serosa. So, moving on to the tunica muscularis externa, it is made up of two layers of muscle. One is the inner circular layer, and the other is the outer longitudinal layer. It is mostly made up of smooth muscle, and between these two layers, you will find the Auerbach's myenteric plexus, which controls the muscle movements. And now, when we talk about the outermost layer, which is here, this is called the tunica serosa, and it can be the tunica adventitia. Now, when we talk about the serosa, it is found in the peritoneal cavity, which means that it only covers the organs which are present in the peritoneal cavity. When we talk about the adventitia, it surrounds the esophagus, rectum, as well as the anal canal, which are not part of the peritoneal cavity. So this is just the basic structure of the digestive tube, or the basic layers of the organs present in the digestive tube. So tunica mucosa, tunica submucosa, tunica muscularis externa, and then tunica serosa or adventitia. Moving on to the esophagus now. Now, when you look at the esophagus from like just outside from the slide or just in your slide box or in Fingerland, this is how they look. This is the human esophagus, and then this is the pig esophagus. And by the way, these are video recommendations for from YouTube, and you guys can watch these to help your understanding even more. So, when we talk about the identification of layers in esophagus, just like we discussed before, the basic layers of the digestive tube, as here you can see, starting from the top, we have the tunica mucosa. Below that is the lamina propria mucosa. Then we have the lamina muscularis mucosae, tunica submucosa here, and then you have the tunica muscularis externa. Same in pig esophagus, tunica mucosa, lamina propria mucosa, tunica submucosa, and then tunica muscularis externa, and then this is apna tunica adventitia. So there is that. And then when we specifically talk about the tunica mucosa, right? So uh, when we talk about the tunica mucosa specifically, it is composed of three different layers, as I said before: the lamina and the laminar epithelium, the lamina propria mucosa, as well as the lamina muscularis mucosae. So now it is important to remember, first of all, when you talk about the epithelium, it is stratified squamous non-keratinized epithelium. Now I have attached the zoomed pictures of this as well. So as you can see, the cells at the top are flattened. This basically means that they are stratified squamous non-keratinized epithelium, because if it was keratinized, you would see a dark layer or a layer of dead cells, you know. So there's that. And then when we talk about the lamina propria mucosa, it is made up of loose connective tissue with different blood vessels and nerves. So this is as you can see, this is loosely arranged, loose connective tissue. So obviously that is uh, lamina propria mucosa. And then when we talk about this layer specifically, this one, this is called the lamina muscularis mucosae. So now the special thing about the esophagus is that it usually you have two layers of muscles, the inner circular and the outer longitudinal layer in the lamina muscularis mucosae. But in esophagus, what you have is that there is only one prominent longitudinal layer of skeletal muscles, as it says. So this is one of the differential diagnostic features for the esophagus as well. So, so just overall to summarize, stratified squamous non-keratinized epithelium, lamina propria mucosa, and then you have only one longitudinal layer of the lamina muscularis mucosae. What this means is that you can just identify the slide of esophagus just by looking at the tunica mucosa layer. Another additional feature of the Another additional feature of the esophagus is that sometimes in the tunica submucosa you can find some mucus glands present in the neck. However, this is not always present, so you cannot consider this as a differential diagnostic feature. All right. But it is important to remember these mucus glands, they are more prominently present in the duodenum. These are called the Brunner's glands. So moving on, so the layer would be the tunica muscularis externa. Since we know the esophagus is a very long organ, it is around 25 centimeters. That is why the type of the muscle in the tunica muscularis externa changes. So in the initial one third, both layers are basically made up of skeletal muscle. In the middle part, there's mixture of both the skeletal muscle and smooth muscle, which basically means in the middle part there is a transition from the skeletal muscle to the smooth muscle, and then the lower two-third is basically made of the smooth muscle. How do you know this now? When we look here, tunica muscularis externa. For example, as you can look here, yeah. So the upper part, the nuclei are essentially elongated, as I have attached a zoom picture of this here as well. So as you can see, the nuclei are elongated. What that basically means is that it is smooth muscle. And then as we can see here, the nuclei are present at the periphery, uh, and this basically suggests that this is a skeletal muscle. So now this shows that there is the transitioning happening between this smooth muscle layer and then the skeletal muscle layer. So that basically means that this is the middle part of the esophagus. However, when we look at this part, you can only see that the nuclei are essentially present at the periphery, which basically means that this is only the skeletal muscle, and hence this shows the upper one third of the esophagus. So that was about the tunica muscularis externa. Now, when we talk about the tunica adventitia and tunica serosa. Now, first of all, What's the difference between both of them? Both of them are surface coverings, but it is important to remember that tunica adventitia, it is not limited by any boundary, and it continues to the surrounding loose connective tissue. However, when we talk about the tunica serosa, it is limited by the peritoneal cavity or the structures which are present in the peritoneal cavity. So only a very small part of esophagus is covered by serosa, and mostly the esophagus is covered by tunica adventitia. So again, when we talk about it, it is made up of loose connective tissue, adipocyte, vessels, and nerves. And then it is important to remember that between the muscle layers of the esophagus, yeah, there's a chance that you might see these nerve bundle structures. These are called mesenteric erbox plexus. Now, what do they represent? These represent a vegetative autonomic network of nerve fibers. I know that may sound confusing, but it is just that these are made up of unmyelinated nerve fibers. That's it. It is a network of unmyelinated nerve fibers. Now, when we move on to the slide of the stomach, so this is the slide of the stomach. You can see these two, and this is the slide of duodenum. The reason why I put the both of these slides together is because most of the people they make mistakes while they're diagnosing these slides. Because as you can see here, the stomach slide, it looks like it has a very thick tunica mucosa, and even here you can see it looks like duodenum has a very thick tunica mucosa, which makes people like confuse both of these slides. But it is important that you zoom in the slides and look for specific structures, and not just look at the slide from your naked eye. And yeah, but it is good to have a basic. I'm not saying that it is bad to have any idea before, like looking at the slide by your naked eye, but you should not just trust that. You have to zoom in and be 100% sure of your answer, because you only get like one chance. So when we talk about stomach, from a gross anatomical point of view, it has four parts. We have the cardia, the fundus, the body, and the pylorus. But histologically, and in your test, you will only have to identify the fundus or the body, and there's not much difference between either of them, so it is easy to identify. So now, one main thing which you see here in the tunica mucosa are these gastric pits, or these are called foveolae. All right. So these are lined by the simple columnar epithelium. As you can see here, zoomed in, you can see these elongated cells. It is lined by simple columnar epithelium. However, the main thing is to remember that we have two different types of cells which are essentially present in this gastric pits, and these release a special type of mucus which protects the surface of the epithelium and the underlying structures against the acidic pH of the stomach. What this basically means is there are cells present here which basically release mucus and which protect the lining epithelium of the stomach from the acidic pH which is present inside the stomach, obviously due to the HCl. And never confuse these mucus secreting cells with goblet cells. There are no goblet cells as present in the lining of the stomach. All right. When we talk about the lamina propria mucosa of the stomach, it contains loose connective tissue, and the gastric pits, which are basically present here, right? They act as the openings of the proper gastric glands. Now, what are proper gastric glands? It is not labeled here, but you see this part, yeah. This basically shows the opening of the proper gastric glands, and these proper gastric glands here, they are always, they are interposed along throughout the entire lamina propria mucosa, and they're made up of mainly two cells. These are called the chief cells and the parietal cells. Okay. And then when we talk about the lamina muscularis mucosae, again, it's the same case. There are two layers of muscle, inner circular layer, outer longitudinal layer. So now when we look at specifically into the two cells, which are the chief cells and the parietal cells, so marked with these arrows, white arrows, these are the parietal cells, and these blue areas or these dark cells, these are called pepsinogen cell. These are called chief cells, sorry. And here is a screenshot from your lecture as well. So as you can see, parietal cells, they are lighter and they are bigger compared to, chief cells. Chief cells are darker and as well as basophilic. Okay. So now the main function of the parietal cells. They're involved in the production of hydrochloric acid, and they're also involved in the production of bicarbonate ions. Now, what's the purpose of producing bicarbonate ions? It is obviously in the formation of the mucus blanket, and what does it do is that it is involved in the neutralization of the pH of the blood. So, it does not allow the epithelium of the stomach to get destroyed by the acidic pH. All right. And moreover, it is also involved in the production of the intrinsic factor, which binds to the vitamin B12 in the lumen of the stomach, which is an important protein in binding and transportation. Oh, sorry, I messed it up. So it is involved in the formation of the intrinsic factor, which basically binds to the vitamin B12. And once the intrinsic factor and the vitamin B12 they join together, yeah, they form a complex which is absorbed by the cells of the ileum, which is involved in the formation of erythroblast. And when we talk about if this complex is not formed, this results in anemia, which basically means lack of blood cells. In case I mess that up again, I will explain again. What does this do is that parietal cells they release a substance called intrinsic factor, which binds to vitamin B12 in the lumen of the stomach. And then this complex of intrinsic factor and vitamin B12 is absorbed by the epithelial cells of the ileum and leads to the development of the erythroblasts. Now, when you talk about chief cells, they have a different functions. Chief cells, they basically produce pepsinogen and lipase, and they produce pepsinogen, which is in an inactive form. Why? This is because pepsinogen degrades protein. So if it is a protein, so if it is released in an active form, right, it will lead to autodigestion and destruction of the whole epithelium. That is the reason why it releases the pepsinogen in an inactive form. Remember, guys, professors might ask you this whether it produces pepsinogen in an active form or inactive form. So make sure about that. However, it releases the chief cells, they basically release lipase in an active form. It's already active, so it does not require any other steps. Now, when we talk about the layers of tunica submucosa, tunica muscularis externa, and tunica serosa, there's not much difference. Tunica submucosa, it again contains loose connective tissue, vessels, nerve, and also submucosal plexus of Meissner. Now, when we talk about the tunica muscularis externa, uh, this is a bit different and it might not be different as well. So it is made up of, 2 or 3 layers of smooth muscle. Now, first of all, here you can see, these here around this area, you can see some oblique bundles. This is incomplete layer, which is only present in the anterior and the posterior faces of the gastric body. That's it. And then you have the properly arranged layers, which is a circular layer, which is reinforced into the pyloric sphincter. And then you have the outer longitudinal layer, obviously. And yeah, and then again, as I had mentioned before, for the esophagus between the muscle layers, there's a mesenteric airbox plexus. So there is that as well. Again, network of unmyelinated nerve fibers. And lastly, the tunica serosa. It was not on the slide in Fingerland. I don't think so. Yeah, you cannot see here because of the staining and the cutting of the slide. So tunica serosa, again, same composition, just made up of loose connective tissue, blood vessels, and it acts as the outer covering of the stomach. So that's about it for the esophagus and the stomach.
Hello everyone, I'll be talking about the duodenum, but before we jump into that, let me tell you a few things about the small intestine. The small intestine has a few modifications, the first one being something called as the Gurking's valves. They are made up of the mucosa and the submucosa, and it is mostly seen in the lower half of the duodenum, entire jejunum, and the upper part of the ileum. The next is something called as the intestinal villi. They are most numerous in the duodenum, and they are just made up of the simple columnar epithelium and lamina propria. The next we have something called as the microvilli, and they just form a brush border. And then we have crypts of lubricant. They are glands which are lined by a simple columnar epithelium. So this is a slide of the duodenum, and it also has like the 4 basic layers: tunica mucosa, submucosa, muscularis, and tunica adventitia or serosa. So there are a few modifications about the duodenum which you have to know. This one thing is called as a Kirkring's valve. Like, let me tell you over here better. So this is one Kirkring's valve, this is the second, and so on. So the Kirkring's valves of the duodenum, they are lower and wider. And next we have the intestinal villi. So these slender structures, this is called as the intestinal villi. So this is one villi, this is the second, this is the third. So in the duodenum, they are like a lot of them are present. They are slender and they have like a characteristic leaf shape. The third thing is that it has microvilli on the enterocytes, but then you can't really see it over here. You can only see it in electron microscopy. So the first layer, that's the tunica mucosa. It is lined by the simple columnar epithelium with enterocytes, goblet cells, and enteroendocrine cells. The next, like this part, like let me tell you, like this is an enterocyte, this is a goblet cell, and this also is a goblet cell. Below it, we have the lamina propria mucosae. It is made up of loose connective tissue and the crypts of lubricant. The crypts of lubricant are made up of these five cells: the enterocytes, goblet cells, enteroendocrine cells, stem cells, and Banach cells. So, the enterocytes, their main function is to absorb substances such as vitamins and everything which is present in the food. Next, we have the goblet cells, like these are goblet cells over here in crypts of lubricant. Their main function is to secrete the mucinogen, which protects the lining of the intestine. We have the enteroendocrine cells next, and they are mainly present in the duodenum, and they produce enzymes for the regulation of the intestine. We also have the stem cells. They replace the entire intestinal epithelium, and they have like a lifespan. Like for example, some cells will be replaced within 2 to 3 days, and like so on. We also have the Banach cells. Their main function is to protect the intestine from the microorganisms, and you can't really see them that well in the duodenum, but they're present in the other parts of the intestine. The last layer which we have in the tunica mucosa is lamina muscularis mucosae, and it is made up of two layers of smooth muscles. The outer layer is made up of the longitudinal orientation, and the inner one is of circular orientation. So, right after the lamina muscularis mucosae, we have the tunica submucosa. This entire part is your tunica submucosa. It is made up of loose connective tissue, blood vessels, and a vegetative nerve plexus, which is called as the Meissner's plexus. So, as you can see, like most of it is filled with like these pure mucous glands, and these are called as Brunner's glands. This is a very important differential diagnostic thing which you have to know about the duodenum. So if you ever get a slide which has like a lumen and you can clearly see like it's got a lot of Brunner's glands and the typical Kirkring's valves, then you cannot be wrong. Like it is going to be the slide of the duodenum. These Brunner's glands, they secrete their mucus secretions, and they are emptied into the crypts of lubricant. The last two layers of the duodenum are pretty basic. So we have the tunica muscularis, which is two layers of smooth muscle. This is the first layer. This is the second layer. The first layer is of circular orientation, and the second layer, the outer layer, is of longitudinal orientation. And over here, you can find the Orbach's plexus. Next, we have the tunica serosa and or the tunica adventitia. Like this is the tunica serosa and adventitia. So the tunica serosa covers most of the duodenum except for the retroperitoneal part, which is covered by the tunica adventitia.
So now we're going to talk about the small intestine. So there are certain features and characteristics, as you guys already know in histology. We use specific characteristics that we know are only available in specific slides to basically help us identify where we are. So these are the most important characteristics that are going to help you guys identify that you guys are in the small intestine. First things first, the epithelium that is found in the small intestine, we refer to it as enterocytes. What it actually is it's simple columnar absorptive epithelium with goblet cells, seen here, for example. So, simple columnar epithelium with goblet cells, which are these white cells. It's also known as absorptive epithelium or enterocytes with the goblet cells. Once you see this sort of epithelium, you know that you are in the small intestine. Now, some other characteristics that are very important. Number one, you have Kirkring's valves. A lot of these characteristics, they're modifications that are there to increase the surface area of the small intestine. So these are Kirkring valves. This is Kirkring valve. Here's another Kirkring valve. This is maybe a Kirkring valve, but these are Kirkring valves. And these valves, they are macroscopic structures. They're composed of the mucosa and the submucosa, and they are more prominent in the jejunum. We're going to see the ileum next, and there you guys are going to be able to clearly see the difference between the Kirkring valves here versus the ileum. Here they're much more prominent, and they increase the surface area about 3 times. So first you're going to see the Kirkring valves. Next you're going to see intestinal villi. So if we zoom in, you're going to see here's one villi, here's another villi, here's another villi, here's another villi, this is a villi. Here we have numerous villi, and these villi are the most typical diagnostic feature of the small intestine. They increase the surface area about times 10, and they're composed of simple columnar epithelium, basically the simple columnar absorptive epithelium with the goblet cells. You also see microvilli, which they make up the brush border, as you guys already know. They are extensions of the surface plasma membrane and cytoplasm, but very, very important are the crypts of Librechen. So these are the crypts of Librechen. These structures that are found at the base of the villi, these are crypts of Librechen. And these are simple tubular heterocrine glands. They extend down from the base of the villi to the LPM, and they are lined by simple columnar epithelium. What is very important for you guys to know is that these crypts of Librechen, they are composed mainly or mostly, the two cells that you guys need to know are stem cells and Paneth cells, and these are found only in the crypts. So once more, the differential diagnostic that lets you guys know that you guys are in the small intestine are the absorptive enterocyte epithelium that you guys are going to see, the intestinal villi, the crypts of Librechen with the Paneth cells. So these very eosinophilic structures, these very highly cut, like pinkish structures, these are the Paneth cells, and the Kirk-ring valves. Last time, what you can see as soon as you put the slide in, least magnification, is you see the Kirk-ring valves. You see Kirk-ring valves. You're going to zoom in. You're going to see villi. You're going to zoom in even more. You're going to see absorptive epithelium and enterocytes. You're going to see crypts with the Paneth cells. Boom, you know that you are in the small intestine. Now, these characteristics are found in the jejunum and in the ileum. However, they are at different ratios. You're going to see these in the ileum, but they're not just going to be as prominent, or they're going to look different. Now, for your exams, it's important that you guys know theoretically the cells that are found. You guys need to understand like what type of epithelium is composed, are enterocytes composed of, which is the columnar absorptive cells with the brush border. Why are they important? What do they do? You guys need to know. They're going to ask you guys about goblet cells. You guys need to know about the enteroendocrine cells that are mainly in the duodenum. You guys need to know really well about stem cells and Paneth cells, which are found in the crypts. Please do not forget, stem cells and Paneth cells are found in the crypts. They're found at the base of the crypts. Well, stem cells are found in the basal half of the crypts, yes. And Paneth cells are located at the base of the crypts, okay? And the Paneth cells are red stained. They're eosinophilic. You can easily see the Paneth cells. These are very important for you guys to know. Now, if we were to take a look at the ileum, you guys can see it's going to look very, very different from the jejunum. But let's try and follow like the same pattern. So first, let's look at the Kirk-ring valves. What do we see? Do we see any Kirk-ring valves? Well, they're here. They are present, but they are very rare, inconspicuous. You can't really see them well. In fact, you might just say you don't even see them, but yes. So this is one major difference between the jejunum and the ileum. In the jejunum, you can really see the Kirk-ring valves, but in the ileum, you really can't see any Kirk-ring valves. Next, let's zoom into the villi. Look at the shape of this villi. Now let's look at the shape of the villi in the jejunum. Look at this. So in the jejunum, we see numerous villi and their slender finger-like shape. But when we look at the ileum, look at the shape of this villi. It's much wider, it's club-shaped, and there is less numerous villi compared to in the jejunum. Look at all of this villi compared to the ileum. So we know that the villi are wider in the ileum, they're club-shaped, they're less numerous, and they represent the most significant structure in the ileum. They're very different from the jejunum. You will also see crypts in the ileum. Same as in the jejunum, you might not see the Paneth cells too well, but you will also see crypts, and you know that they're going to have Paneth cells on the base. Now, the most important thing about the ileum is this. The ileum has extensive lymphatic infiltration. And this lymphatic infiltration, just once again, for you guys to see how the jejunum looks like. Now, look here at the ileum. You have all these lymphatic follicles, this lymphatic infiltration, and this is known as Peyer's patches. Peyer's patches are basically an accumulation of lymphatic tissue that's in the intestinal mucosa. And this basically destroys the lamina muscularis mucosa, which, as you guys can see, is very present in the jejunum, but here the lymphatic infiltration has completely destroyed it in the ileum. And the Peyer's patches are important because they're basically there, as you guys know, when we consume food, the food that we eat, like you might have some bacteria, might be infected, might have something there. And basically this is the job of like the Peyer's patches. They're there to combat any sort of bacteria, anything that's antigenic that they might come in contact with. So this is pretty much, in my opinion, the number one diagnostic differential feature between the jejunum and the ileum. You are not going to see this sort of lymphatic infiltration that completely obliterates the lamina muscularis mucosa in the jejunum. So once again, just one last time, just from a difference, the first thing that we're going to notice are the Kirk ring valves. We see Kirk ring valves. We know we're in the small intestine. Here, we barely see them. We are going to zoom in and we're going to look at the villi. Oh my gosh, there's villi. There's so much villi. We know we're in the small intestine. Going to zoom into the villi here. The villi in the jejunum, slender-like, finger-like shape, numerous. Villi in the ileum, wider, club-shaped, less. We're going to zoom in again. We're going to look at the epithelium. Oh, it's enterocytes. It's columnar absorptive epithelium with goblet cells. Again, we know we're in the small intestine. We look at the crypts. We look at crypts. We see Paneth cells, crypts of Lieberkin. We can find them both in the jejunum, both in the ileum. And if we look at the ileum, an additional thing that we see is all of this lymphatic infiltration.